Welcome back. In my last lecture, I described how biotechnology and recombinant DNA have been used to make products using gene expression and vectors, and these products are useful to us in things like medicine, and later on you'll see agriculture. Now I'd like to describe how the organisms themselves can be used to clean up the environment where we have environmental problems. In other words, how these organisms can do things for us after they are genetically modified. My opening story is about landmines. Landmines are a sad legacy of recent history. These mines have wreaked havoc both during and after recent wars in many locations around the world. Landmines are cheap. They've got plastic casings filled with an explosive such as TNT. TNT stands for trinitrotoluene. That's the end of trinitrotoluene. From now on, it'll be TNT, which is the explosive. And these mines are triggered to explode whenever they're disturbed, vibrated in some way. The United Nations estimates that there are over 100 million unexploded landmines lying on or below the ground in countries ranging from Angola to Cambodia to countries in Europe as well. This makes plowing fields or for farming or other uses a risky business. If you're a farmer and you want to use your field after a recent war, plowing it or digging it up and planting seeds is not going to be a thing you want to do if there are landmines in the area. So you'd like to detect them. It's very important. You like to detect them and then explode them or get rid of them some other way. Now, these mines are encased in plastic, so they avoid conventional metal detectors. The best way to find landmines, unfortunately, is the most dangerous. A person gingerly walks around poking the ground with a long stick, and if they hear a mine about to explode, jump out of the way. Not a terrific occupation, and a very dangerous one. At the University of Tennessee, Neil Stewart is trying to use plants and biotechnology to remedy this ecologically adverse situation. This is an example of bioremediation, the use of biological organisms to improve the environment. What Stewart is trying to do is to use biotechnology to make plants that will tell us where landmines are located in a certain region. Specifically, he's trying to make a plant biosensor. Now, the biosensor is going to require two things. First of all, it's going to require a gene that makes a protein that's detectable. That is, he's going to have a plant growing in the ground, and that gene is going to make a protein we can see. And secondly, that protein should be under the control of a promoter, and that this promoter would be expressed when landmines are around. Now, how are we going to get those two things? Well, the 1990s saw the isolation of the gene and the promoter that might be useful. And biotechnology allows us to put both the gene and the promoter into a plant. First, the gene whose product we can see. In the deep sea, there are some jellyfish that glow in the dark because they make a protein that fluoresces. Now, fluorescence means that a chemical absorbs energy, electrical energy or light energy, and then emits that energy as a flash of light. Fluorescent light, for example, emits energy in the presence of electrical energy in the form of light. And here we have a fluorescent protein that fluoresces green in the jellyfish and emits its fluorescence in the presence of the very small amount of high energy light, ultraviolet light, that reaches the ocean depths. It's kind of like the ink that is stamped on my wrist when my students take me to one of their favorite nightclubs in Hollywood, and you stamp the thing on your wrist saying, "You're okay." And they shine a black light on it, and it glows, and the letters "okay" are flashed, and I get by the bouncer into the nightclub. Right. In the 1990s, the gene for this green fluorescent protein made by the jellyfish was isolated. And biotechnologists started putting this gene into vectors to use it just like antibiotic resistance would be, right, for the presence of the vector when it's in a cell. So you'd put the gene into a vector as a marker for the presence of recombinant DNA in host cells. And the result was glowing organisms or cells. Glowing fish at one time, for example, were sold as a novelty. They were taken off the market because people didn't like it. I use, for example, a vector that expresses green fluorescent protein in teaching, and teaching students who are even non-majors, as you are, those who are listening or watching this series of lectures. I teach a laboratory where we will take bacteria and have them make the green fluorescent protein. And it's quite a sobering exercise for a non-scientist to realize that they've, in a very simple experiment, put a gene for a jellyfish into a bacterium. So that's our gene that we're going to be able to see. And hopefully the plant then will fluoresce green. Second, we need a promoter. And we need the promoter that will turn the gene on in the presence of TNT. In the 1980s, microbiologists found a bacterium called Pseudomonas putida, interesting name, that had genes for enzymes that will break down TNT and use its breakdown products for their own purposes. There are bacteria out there that will make genes to break down almost anything in nature. We don't have a lot of those genes. There are certain species of bacteria that do. Into the 1990s, these scientists looked at the genes coding for these enzymes responsible for this metabolism, for this breakdown of the TNT, and they found that these genes, not surprisingly, had promoters that became active for turning on the gene in the presence of TNT. Very interesting. They were TNT-sensitive promoters. You can envision the TNT binding to a protein, and it does, that goes to the promoter and turns the gene on. In fact, just being near the TNT in the soil, in the air, was enough to set off these genes. They were that sensitive. So, now we had the promoter, TNT-sensitive promoter, and the DNA coding for green fluorescent protein. Stewart then took these two together, the promoter and the gene, added them to a vector, and made some transgenic plants. The result was, whenever these plants are around TNT, these plants will glow under ultraviolet light. A biosensor is born. Now, life is not that simple. Challenges remain for this to be a feasible technology in regions of the world where there are a lot of landmines. Well, first of all, you have to spread the seeds around the field and have them grow. Well, that's not a big deal. You can actually do that from an airplane. The seeds will sprout. The transgenic plants will grow, and they do express the green fluorescent protein. But you know, walking around the field with a black light at night, like we would at the nightclub, right? Finding, do you have a wrist that glows? Finding, oh, is this a glowing plant? There's TNT around. I think that's not what you want. It's just as dangerous as poking the ground with a stick. So Stewart and his colleagues are proposing using what is called remote sensing from airplanes that can detect ultraviolet light. And we have many examples in our defense industry of using remote sensing, and this might be combined with seeding from airplanes. So this technology moves along. I bring this up a story as an example of bioremediation. Again, using organisms to solve environmental problems. There's an old biological saying that I learned when I was an undergraduate, and that is, big things have little things upon their backs to bite them, and lesser things still lesser things, and so ad infinitum. Another way of looking at this is the great food chain in ecology, right? Plants are eaten by animals, we eat the animals, and other creatures will eventually eat us, right? Genes of parasites and hosts and energy going through a food chain. knowledge of the food chain allows biologists to use it. We use it in agriculture, of course, but we can use it, for example, to introduce pests of pests. One of my first jobs as a biologist was when I was an undergraduate. I worked with the agriculture department, and my job was to describe the life cycle and ecology of tiny insects. And these insects, related to wasps, only they're extremely tiny, make their living by reproducing inside of pupae, which are the immature stages, or caterpillars, which are other immature stages, of a bud moth, of the apple bud moth in my case. Now this particular bud moth, the apple bud moth worm, the caterpillar, eats up the leaves of apple trees and causes significant damage to the apple trees such that the yield of apples goes way down. If they don't have leaves, trees are not going to produce many apples. The objective was to try to, and we did, introduce this insect, this lesser thing, that would bind on this lesser thing, this bud moth, to introduce these insect pests to reduce the populations of the worm. It would reduce the need for pesticides, and the pesticides weren't working well anyway on this particular caterpillar. This is called biological control. Now it has a genetic basis. The host worm, the caterpillar, has genes for molecules it happens to make for its own purposes. Some of them are defense molecules. They go up in the air, and the parasite, this small wasp-like insect, has glands that detect this worm. And the parasite flies around and says, "Oh, dinner!" And it goes over to the worm, lays its eggs, and reproduces, thereby killing the worm. So there are genes for both the host and the parasite that have been well described. It's a sensor system. Biological control like this is now widespread and works along with traditional ways of pest control using pesticides in much reduced amounts in a process called integrated pest management. We can use bacteria as nature's recyclers. Bacteria eat things, just like our insect that I just described eats things. Bacteria eat things as well, and they thrive on all sorts of nutrients, including things that we refer to as wastes. There are species of bacteria that have the genetic capacity to produce, as I've mentioned, enzymes and therefore biochemical pathways that humans don't. As an example, composting. A compost pile uses bacteria to break down the carbon-rich stores such as cellulose, which is the cell walls or indigestible parts of wood chips or paper or straw or hay, as well as nitrogen-rich sources such as protein wastes, coffee grounds even, right? What's a coffee ground? A coffee ground is ground up coffee seeds. That's part of a coffee plant. And some of that are things that we don't digest, that don't form part of coffee. And so these grounds contain the cell walls of plants, which contain cellulose, and the seed coating of the coffee seed. And those are indigestible, except, believe it or not, by some bacteria. Fruit and vegetable scraps, also digestible by bacteria, even though we, as I mentioned before, can't digest them. So in a compost heap, what happens is the bacteria work on these things and produce four things: carbon dioxide and water. That's the end products of the respiration. So the bacteria are thriving, they're getting energy out of it. And heat. If you notice, a compost pile gets pretty warm because the energy comes off. Remember thermodynamics, right? Energy is neither created nor destroyed. So the energy from these wastes ends up in the bodies of these bacteria, some of it comes off as heat. And then there's indigestible parts even the bacteria can't get, and that ends up as what is called humus. And humus can be spread over the soil to provide nutrients for other bacteria that are in the soil. So these are bacteria doing a job for us, right? They're digesting things to simpler components. Wastewater treatment. Wastewater treatment uses bacteria to act on human wastes and paper products and household chemicals. The liquids and solids here are treated differently. There's one group of bacteria that digests harmful substances in the solids of our wastes. The remaining sludge that they don't digest is used for landfills, to fill up landfills or as fertilizer. Some of these bacteria, by the way, that digests human waste make a gas called methane gas that you may have heard of as a byproduct, and this gas is used for energy. Liquid and solid wastes that solids are dissolved in the liquids are digested by still other bacteria. There's a whole series of bacteria now that will digest different substances that are in things that we call waste. Give you one example is ammonia, which is a toxic thing that is in urine. Ammonia, up until now, has been digested by a group of bacteria that are in the wastewater treatment plant, and is digested to various molecules, ultimately to nitrogen that goes up in the air as nitrogen gas. You'll recall ammonia, amine groups, are breakdowns of proteins. Toxic to us, ammonia, but not to the bacteria who different bacteria break it down in different steps. Recently, a single bacterium, a new species was discovered called Candida brocata ammonooxidans that will break down ammonia to nitrogen in a single step. And this is now going to be used widely in wastewater treatment. So we're still discovering more efficient and better ways to use bacteria for our purposes. This is bioremediation in wastewater, for example. Bacteria and plants can be given or have genes that remove pollutants. So in addition to being nature's recyclers, bacteria break down many, many human-made pollutants. Well, how do we discover this? We can discover this ability by bacteria in the following experiment. We can take some soil or water, with water, and we take the pollutant that we're interested in, like oil, and then we look and see whether any of it gets broken down. And those bacteria that live in the presence of that pollutant are the ones that are thriving on it. And then we grow up those bacteria and we say, oh, that's a bacterium that likes to break down this plastic or this oil or whatever. Okay? For example, there were bacteria that were isolated first from soil. In this type of experiment, we could add oil to them and see whether the bacteria would thrive, that were essentially isolated on each component of oil by a scientist named Ananda Chakrabarty in the 1970s. Now what Chakrabarty did was contaminate soils separately with all the different major components of crude oil. And there are about a dozen of them. So you take component number one, chemical number one, he'd add it to the soil and see whether there are bacteria that eat it. And then component number two and component number three. So you had a whole bunch of bacteria now that would break down each component of crude oil. Then he did bacterial genetics at the time, this is the 1970s. He did bacterial genetics and mated them. This is before gene cloning was really invented, just the beginning of it. And so he did traditional bacterial genetics to get a single genetic variety, a superbug, we call it, that would break down multiple substances, a whole bunch of different components of crude oil. Now, Chakrabarty was working for a company, so he applied for and was awarded a patent for this superbug in 1980. This case was argued for a number of years by the U.S. Supreme Court. And the Supreme Court ruled, and I will read, that a live, human-made microorganism is patentable subject matter under Title 35 U.S.C. 101, that's legal stuff. Respondent, Chakrabarty's microorganism, constitutes a manufacture or composition of matter within that statute. This was a landmark decision by the Supreme Court. Essentially said a genetically manipulated organism could be patented, and led to a flood of patents for genetically modified organisms as well as genes. Now you'll recall in a previous lecture I described how the scientists Cohen and Boyer and their universities who invented the method of making recombinant DNA patented it and reaped great rewards from it. Now we're seeing things like patenting genes that people are finding. Every university now of substance has a patent office, and they are busily patenting every gene and every process that biotechnologists find in the hope that they'll hit a home run and get something valuable. I'll say that very few home runs have been hit, as is true in baseball. There have been a lot of strikeouts. One example that's going on is that, for example, the patent for human embryonic stem cells was filed by the university where it was discovered, University of Wisconsin, and that is being argued now in lower courts because scientists are worried that it could limit the research that people could do on stem cells because they would have to pay expensive royalties for any experiment they want to do. In 1989, the oil tanker Exxon Valdez ran aground near the Alaskan shore, releasing 11 million gallons of crude oil over 1,000 miles of shoreline. This was an environmental disaster of major proportions. Cleanup by physical methods such as skimming the water and spraying the rocky shore with detergents was used first, and the result, this dispersed about 2/3 of the oil. Bacteria did the rest by bioremediation. These genetic strains that can eat up oil that Chakrabarty and others had patented. Nitrogen and phosphorus salts were first sprayed on the shoreline to get the bacteria to thrive. So it gave them a nice growth medium because they normally don't live on oil. They need other things. And then the Pseudomonas bacteria which were already there would grow, and other strains were added so that all of the oil would be digested. And they worked very quickly to digest most of the remaining oil. This process is ongoing. The government of Kuwait is using bioremediation to try to clean up 250 million gallons of oil, way more than the Exxon Valdez, 20 times more, that was spilled, probably deliberately in a number of cases, by exploding oil wells, onto the desert during the Gulf War of 1991. This is probably the largest single bioremediation project in the world, and it's going on as I speak. There is a type of bacteria called extremophiles that have many, many genes that are useful in bioremediation. What are extremophiles? Now, phile means you like, right? Extreme means like. These are microbes, bacteria, that love the extremes of nature. They're kind of the ultimate, right? The ultimate athletes of the biological world. These are bacteria, usually, that can live in very hot places like hot springs, or the ice in Antarctica, or high pressure deep in the ocean, or high salty, very salty environments like what we used to think of the Dead Sea. In fact, these organisms form a separate group of organisms called the archaea, a word for old. And they're called archaeans, because they resemble the organisms that some scientists believe are the first living cells that were on the earth. The archaea genomes have been sequenced, and half of the genes of the archaea do not resemble anything in other types of bacteria or eukaryotes. They're prokaryotic cells, but they are real different. For instance, some of them have genes that use carbon dioxide, just like plants do, not to make sugars, but to make methane gas. And archaeans are by far the major producer of this methane gas. It's one of the greenhouse gases. The granddaddy of all archaeans is a bacterium called Deinococcus radiourans. Deinococcus radiourans is a bacterium that lives in probably the most dangerous environment of them all, extremely high levels of radiation. Now, normally, radiation kills cells by damaging DNA. And when when a human's or any cell's DNA is damaged by radiation, we can repair it. We have a system of repairing the type of damage that radiation does to DNA. But large amounts of radiation overwhelm that, and you get, as we've mentioned before, permanent mutations and cancers as a result. Now, Deinococcus radiourans gets around this by having the most efficient and sensitive radiation repair system in nature. It's a phenomenally good system, better than any other, way better than we do. Now, genes from other extremophiles that live in high salt, high temperature, etc., are being engineered into Deinococcus radiourans. And so no matter what the environment, this microbe keeps coming back. People call it Conan the bacterium, after a movie called Conan the Barbarian. It's used to clean up the most toxic sites we know of. For example, in America, there are these Superfund sites, which are a group of sites around the world that are extremely contaminated with extremely bad stuff. And these Deinococcus bacteria that have all of these genes for cleanup are being used there. Plants can be genetically engineered for environmental cleanup. For instance, bacterial genes that allow environmental cleanup can be put into transgenic plants to break down oil. There are plants that will convert solid mercury and arsenic into harmless substance. Now you might ask, well, why use plants when microbes are available and around? Well, the issue is you want to get the microbe out of the soil when you don't need it anymore. You don't want extra microbes. It upsets the ecology of the soil. And getting bacteria out of the soil and the environment is quite difficult once they're established. Plant is easy. You plant it, it does its thing, then you take it out, you harvest it. So that becomes a lot easier. So this might be a better way of doing bioremediation in some cases than bacteria. Mining is another industry that is using bacteria, some of them genetically engineered, to extract metals. Mining for minerals is an old human activity, and it's changed very little over the millennia of time that humans have been mining. You dig up the ground, ores are taken out. Ores are complexes of our mineral of interest with other things. Then the minerals like copper and gold are extracted from the ores. But this requires, to extract the minerals, very harsh methods, such as really toxic chemicals and a lot of heat. You may know that mining is environmentally a very damaging process. There's a bacterium called Thiobacillus ferrooxidans. This bacterium uses a chemical called copper sulfide to get energy. We use carbohydrates to get energy. We break down sugars to get energy. We convert them to CO2. In these bacteria, copper sulfide, which is an inorganic chemical, is used for energy. What happens? They use it and then they release copper. Well, that's what we want in mining, isn't it? And so now what is happening is the ores are extracted, the complex, are sprayed with sulfuric acid to make copper sulfide. The bacteria come in, the bacteria then grow and leach out and give us the copper. This now accounts for about a quarter's worth, $1 billion of all copper mined in the world. Bacteria are doing some of the mining for us. Problem with this is the bacteria, and they do this, they grow very rapidly. They release a lot of heat. It's a breakdown product of metabolism as they break down the ore, and this slows down the process. So scientists are now using recombinant DNA technology to transfer the genes for the release of copper into other extremophile bacteria from a hot spring. And these extremophile bacteria in the hot spring live in hot environments, so they can tolerate the heat. So these bacteria will be heat tolerant and able to break down the copper to release it for our use. As we will see in the next lecture, the bacteria in hot springs figured prominently in another story of DNA manipulation, development of the polymerase chain reaction. Thank you.